Hi, everybody, and welcome to our conversation from the front lines today at Congregation Shereth Israel. I'm Rabbi Jessica Zimmerman Graf, the senior rabbi, and joined by Dr. Jonathan Graf and Dr. Jeremy Rassen. It gives me great pleasure, although I can't, let's see, why can't I see Dr. Jeremy Rassen? I don't know, hopefully he'll pop back up.
I can see myself here.
He's right there. He's sitting underneath you. That's why you can't see him.
Outstanding. It gives me tremendously great pleasure to welcome you back to Congregation Shereth Israel. Jeremy, for those of you who don't know Jeremy and the Rassen family, Jeremy grew up here and tormented me for years in Sunday school and Hebrew school. But I have to say, I think learning how to ask questions about the weather in Hebrew and meeting Jeremy were the two best things that came out of our childhood education here at Congregation Shereth Israel.
And I had to move east so that I could be somewhere where I could say Yesheleg Bachutz.
Awesome. That's what we did in Hebrew school all the time. We talked about the weather. That's outstanding. So I want to welcome you all. When I invited John and Jeremy to talk about this topic of vaccines and COVID as part of our series on From the Front Lines and what's going on in the medical world in our crazy time, it was a very timely topic. And oh boy, we didn't even know how incredibly front and center this topic would become. So I want to thank you both so much for being with us to talk about this today. And I'm going to turn it over to John to start. And then by way of introduction, you know what? I know a lot about you, but I didn't even print a bio. Jeremy was probably the smartest kid in Hebrew school, so I'm not surprised that he went on to get degrees in computer science and public health and start a really amazing company that studies the way that vaccines and medications work out there in the world using huge amounts of data from the CDC and other places. Is that sort of accurate? You can introduce yourself.
I'll
Here we go.
There were many, many, many smart kids in Hebrew school, for the record. Many, many, many.
Well, welcome everybody. It's a pleasure to be here again. We took a little hiatus for the last several weeks. We had high holidays and other events intervening, but we're back. COVID is certainly back. If you guys have, of course, been reading the news lately, both here in San Francisco and pretty much throughout the country, the COVID case counts are up anywhere between 50 and 400% in just the last few weeks. And there's a lot of reasons why this is probably going on right now, not the least of which the weather is changing, people's habits are changing, and basically COVID is now spreading for a whole bunch of different reasons, depending upon which community one is spreading pretty rapidly throughout the country right now. And so there's no more timely time, I think, to talk, to sort of get our hopes up, I think, at this time, and to sort of talk vaccines and virology and immunology. And so that's our plan this evening. I, as a rheumatologist and immunologist, I thought I'd start the session for about the first 10 or 15 minutes, giving a brief overview about vaccines and about which vaccines are in development, and about specifically the vaccines that I think we've all heard about in the news this week that appear to give us great promise. Then I think I'll switch our attention to a one-on-one conversation with Dr. Rassen here. I kind of like how that sounds. Jeremy, not only is a previous...
I have to point out that since at least my mother and perhaps my father are both on the line, that the real Dr. Rassen in the family is my dad.
Every Jewish family wants their son and daughter to be a Jewish doctor, right? So they have one. And in a way, Jeremy's patients are basically, he's not a medical doctor, he's a PhD and an epidemiologist extraordinaire. But Jeremy has founded a company called Aetion that has a proprietary patented computer science technology that basically allows it to analyze data from all walks of life, mounds and mounds and billions of data points, whether it's healthcare claims, government data, hospital records, whatever it may be, can basically take billions and billions of pieces of data and distill them down to answer real world clinical questions to sort of optimize healthcare delivery in the United States. And Jeremy's company's clients include large pharmaceutical companies, the Food and Drug Administration, various government agencies, health policy agencies that will basically hire Jeremy's company to consult with them and to basically do real world rapid data analysis that can provide solutions potentially for companies looking to better function in the current healthcare environment. So what better person to talk to today about vaccines and about what we've learned about vaccines, about how we're going to know if a vaccine's effective or not, and how can we potentially use a vaccine in the most effective way and distribute it throughout the country, than Jeremy, who no doubt, I don't know this to be a fact, but we'll ask him in a few minutes. I'm sure there are many agencies consulting with Aetion right now about how just to do that, how to get a vaccine most effectively into the community to basically beat down the transmission of this disease. So with that, let's get started. For those of you who want to know basically about vaccines, if you've been paying attention to the news at all this week, there have been, or in the last couple of weeks, there have been two vaccines in particular that have made recent headlines. One is a vaccine produced by Pfizer Pharmaceuticals in cooperation with a company called BioNTech, which is actually a German company. And the second vaccine is a company produced in cooperation between the NIH, an institute at the NIH called the National Institutes of Allergy and Infectious Disease, and a company called Moderna. Those are the two vaccines that are actually, we've heard a lot about in the news, and I'll talk about those in a second. But basically, a vaccine, as you know, is a product that's actually administered that induces an immune response that hopefully either prevents one from getting infected from a pathogen, or if one is to get infected in a less serious way, because the body has already prepared an immune response to the vaccine, what's called a primary immune response. And then when the body gets re-challenged by a pathogen, theoretically with the same proteins and structure that the vaccine had in it, the body can mount a more rapid secondary response the second time around, the third and fourth time around, such that a patient either doesn't get sick, or if they're going to gets sick for a shorter period of time and in a less severe way, because the immune response is able to sort of mount itself in a much more rapid way. So you know, just to just sort of lay it out there simply, when we all come across infections for the first times in our lives, it takes the body a good 2 to 3 weeks before it has the ability to mount a very elegant, what we call a pathogen-specific way in which it can fight a particular pathogen, like a bacteria or a virus or whatever. And so during the 2 weeks it takes to get the immune response up and running, people can become very, very sick, and often the infection and the immune response to that infection can get out of control before the body has an ability to sort of attempt to bring the infection under control and to clear it as well. And there are various mechanisms that are in place, whether it's certain parts of our immune system called innate immunity that try to tampen down these responses until the adaptive immune response that's pathogen specific kicks in 2, 3, and 4 weeks later. But often that's not good enough. And in the case of the current coronavirus, a lot of patients get sick usually within 7 to 10 days of being infected, which is before the body can mount an effective immune response to protect it. And by that time, the pathogen has already entered the lungs and we're off to the races. So the idea with this vaccine is to sort of educate the immune system to sort of recognize components of the virus, particularly those components that actually allow the virus to attach to cells inside the body and enter cells and begin the replicating process that the virus undergoes in its life cycle to begin growing and replicating and invading other tissues like the lungs and elsewhere as well. And so the concept of these vaccines is to sort of, again, educate the immune system. At that point, the immune system, then when it does see the coronavirus in the real world, will say, aha, I've seen you before, and will mount a very specific immune response rapidly this time. It won't take 2 weeks. The recall immune response can take a day or 2 days to mount an antibody response, or what we call a cell-mediated response, where cells or antibodies will go and attack the virus or attach themselves to the virus and prevent it from entering the host. And if some virus does get into the host, then there are cells around that recognize a virally infected cell much more quickly and will kill that cell before the virus has had a chance to replicate itself and get released and infect other cells. So that's the idea of a vaccine, to educate our immune system so that when we see the actual pathogen, what's called a recall response, which is much more rapid, much stronger, has memory to it, real memory, kishkas, that can actually go and tampen down the infection before it gets out of control. Now, there are many different ways you can make a vaccine. We got a lot of questions sort of in the run up to this particular talk, asking about what type of vaccines we were talking about in the news today. There are many ways you can make a vaccine. The classical way you'd make a vaccine, let's say if it's polio virus or influenza virus is a good example, is you basically take your virus and you have to grow it. So in the case of influenza, it actually grows in eggs of all things. And so there are companies out there that make influenza vaccine that have these giant bioreactors full of egg products, basically, where they grow in millions of gallons of influenza virus. And then so that takes time. And then you have to go and purify the virus from the egg and process it and mix it, with various things before it becomes an actual product. And so you can either take a whole virus and then the influenza vaccine, then the influenza has to be either treated in some way to render it, not a pathogen, or killed, right, in that manner. That's the influenza vaccine that we all get when we get our shots. Other vaccines can actually be pieces of a particular pathogen, and those have to be also grown up and processed, or they can be done through special kind of what we call recombinant DNA technology. They can be manufactured separately, but all these processes take a lot of time. And in a lot of instances, you actually you run the risk. You have to grow the pathogen, which is dangerous. You have to purify the pathogen. You have to inactivate the pathogen so it doesn't make other people sick. And a lot of this can take a lot of time in development. And this particular way of making vaccines, which is the way we came up with smallpox and polio, and we do influenza and all these other things, doesn't lend itself very, very well to a pandemic, right? In a pandemic, right, a new pathogen emerges, everybody gets sick, you're under the gun, and you don't have 2 or 3 years to wait to develop this kind of a vaccine, let alone another year after the vaccine is to be tested. And then once the vaccine is tested, you have to actually then go and manufacture it, which can take a long time with sort of that old-fashioned technology. And so what's different is the 2 vaccines we heard about this week are nucleic acid vaccines. It's very, very clever. Both the Pfizer BioNTech vaccine and the Moderna NIH vaccine are what are called messenger RNA vaccines. So what is messenger RNA? It begs the question. So all of our cells contain DNA in our nuclei, which is the foundation of, and the way that DNA actually leads to life is that genes are actually turned on. And when genes are turned on, there's a machinery in the nucleus that transcribes the genes. And the DNA gets transcribed very, very faithfully from its code into an RNA code. And then the RNA leaves the nucleus, goes into the cytoplasm of the cell, goes into the broader body of the cell, and then it gets translated into amino acids, which become proteins. And that's where proteins come from inside cells. And so what the 2 vaccines actually, the Moderna vaccine and the Pfizer vaccine have done is they've actually taken messenger RNA from the virus itself that codes for a protein of the virus and have placed it into their proprietary vehicles, their RNA vehicles. And this is really, really revolutionary. Why? It's revolutionary for several reasons. First of all, in order to make an mRNA vaccine, you don't have to grow virus. You can make it, once you know the genetic code, all you have to do is take the RNA code and plug it in like a cassette into the RNA vehicle as well. And so in the case of the Moderna vaccine, when the sequence for the coronavirus was first published in January, within 42 days later, there was a working vaccine candidate. Moderna had its vaccine candidate. And so literally they had a working vaccine within 40 days. In the world of influenza vaccines and polio vaccines and things like that, that's just unfathomable. 20 years of research basically distilled down into 40 days. That's what an mRNA vaccine can do. The other advantage to an mRNA vaccine is when mRNA is injected into the body, it gets into cells, right? It gets into the body of the cell, the cytoplasm. The host cell, your machinery, your translation machinery looks at that RNA and will translate it into the actual coronavirus protein. And then your cells will display that protein on its surface. And so the body will then see a foreign protein on your own cells, think that you're virally infected, right? And mount the primary immune response that we all talked about. It's a very effective way to mount an immune response. And I won't get into specifics, but and finally, there are some RNA vaccines that have the added ability to replicate. The 2 vaccines we heard about this week do not replicate on their own, which I think is a good thing. But there's theoretically advantages to having, there are several companies out there that are making RNA vaccines to the coronavirus that will actually replicate themselves. And so you can give a very, very tiny dose to someone, it gets into cells, and then the cells can replicate the RNA almost in perpetuity on its own to continue making coronavirus protein, which again is a very effective way to mount an immune response. So I'm going to turn to my wife here just for a second to make sure, is that complicated or not complicated? Let me know if I need to simplify or good. She gives me the thumbs up. She's understanding this, which means we're in good shape here. So there are some disadvantages to an RNA vaccine. You want to know, why haven't we done this before? There's never been an RNA vaccine ever that's made it to the FDA approval process. And there are a lot of reasons for this. The classic reason is that RNA, having worked in a lab, can say this with certainty, is very difficult to work with. It degrades very, very easily when it's not inside of a cell. So if I were to inject any RNA into anybody here, it would be obliterated into pieces within seconds probably. There's even an enzyme on all of our hands called RNA Ace that will actually degrade RNA within seconds. So what's amazing about the technology that's coming from these companies is they've found a way to stabilize the RNA so that it doesn't degrade once it gets into the body. They've also managed to place it in sort of a layer of fat kind of particles that allows it to get into cells without degrading. And then that special kind of proprietary cocktail that the RNA is sort of bathed in allows the RNA to be expressed inside of cells and effectively translated into virus protein. And so these are incredible technologies that are sort of out there. And then the last thing I'll say is because these are RNA vaccines, there really isn't this fear. You know, you wonder, well, why haven't we made DNA vaccines, right? Why not make DNA? DNA gets into DNA vaccines, which there are some being made now for the coronavirus, also can get into cells. They have to get into the nuclei. And once they're there, they can make many copies of RNA, not just 1 or 2 copies of RNA, but many ones and amplify a response that way. But there's always the difficulty of getting DNA into the innermost part of the cell, into the nucleus. It's easier to get into the body of the cell than into the nucleus itself. There's also the risk that once the DNA gets into your nucleus, it can be inserted randomly into parts of your own genome. And if it's inserted in the wrong place, you can actually have mutations that lead to cancers and things like that. So there's always been this theoretical fear with DNA vaccines. Those fears are not as much of a concern with RNA vaccines because they don't go into the nucleus and mess with your DNA. So anyway, that's a lot of information out there, I think, to sort of digest. But what did we hear about this week? So the first vaccine we heard about, I've gotten lots of questions. So the short answer to some of the questions I've gotten, is this vaccine a live vaccine or are these vaccines live? No, none of these are live vaccines. And in particular, the 2 vaccines you heard about this week are not live vaccines. They're artificial RNA that's put into a vector that's then injected into people, gets into your cells, and your cells express singular proteins from the coronavirus without expressing the whole coronavirus itself. So it is not a living vaccine. Okay, so what are some of the issues that, so first let's talk about the 2 vaccines that are out there. The first one is the Pfizer vaccine that's made with a company called BioNTech. It's the vaccine BNT162. It was found to be 90% effective. I think we all heard in the news today. And what does that mean? We'll talk to Jeremy a little bit about this in a second. But basically, these are phase 2 and 3 combined clinical trials. And these are trials, I think Pfizer initially was looking at 30,000 patients and has expanded now, I think, to 2 or 3 times that number for their trial. But rather than wait for the first data to come in on 30,000 patients, they have pre-specified interim analysis points. I believe there was one at 300 patients and then another one at about 6,000 patients. And I think it's the 6,000 patients that we're hearing about right now. And there was a 90% reduction in cases of COVID disease and in cases of severe COVID. And we can talk about what that means in a second. The Moderna vaccine that came out today is really an incredible partnership. So the Pfizer vaccine is a partnership between American pharmaceutical company Pfizer and a German biotech company called BioNTech. These 2 companies actually are partnering on influenza vaccine technology using mRNA. And so when the coronavirus came out, they leveraged an existing agreement to develop influenza vaccines and actually agreed to partner on the development of a coronavirus vaccine. So BioNTech has the vehicle, They took the sequence from that was published from the Chinese in the first stages of the pandemic, created this product, and Pfizer now is leveraging its large pharmaceutical backbone, its vast financial and resources and network around the United States in terms of clinical trial apparatus, and they're developing the product together. The Moderna vaccine is actually an amazing product. So Jeremy, still, I believe, you still take up residence a little bit in Cambridge as well, although you're based in New York. I know you have roots in Cambridge and came from Harvard and other places. Moderna is a Cambridge-based biopharmaceutical company. They have the vehicle. The National Institutes of Allergy and Infectious Disease at the NIH developed a construct, an RNA construct for the SARS the spike protein, the S2 protein, and they, in partnership with Moderna, developed what's now the Moderna vaccine, Moderna 1273. And once it went from early phase development, I don't know if you guys heard about Operation Warp Speed, but they then, and this is where I think Jeremy, I'm sure, got involved. There's an entity called BARDA, which is the Biomedical Advanced Research and Development Authority. The government loves large acronyms. And so what basically BARDA is basically the Pfizer in this entity. It's a government-sponsored entity that's, of course, spending billions of dollars to develop this product. They have the clinical trial network that they've set up all over the country, and they're supplying the financing for what's called the COVE trial, which is a phase 3 study that we just heard the interim results from yesterday, as a matter of fact. And so the NIAID has passed this on to BARDA, and BARDA, in cooperation with Health and Human Services and the Operation Warp Speed that the government announced several months ago, is actually putting the financial resources into doing this phase 3 trial and ultimately into getting this vaccine to the public should the results of the phase 3 trial be successful. So what's, I think, remarkable about the government response here, the government's done a lot of bad things and wrong-headed things, I think, during the pandemic. But one of the things I personally think they've gotten right is they've actually spent billions of dollars up front to not only build the clinical trial infrastructure to study vaccines like the Moderna vaccine with a government-private partnership, but they've also pre-purchased doses of the vaccine, both from Pfizer and from Moderna and others, hundreds of millions of doses of the vaccine, not even knowing whether the vaccine was going to work or not, basically basing this on good judgment and on good educated guess that these vaccines were some of the more promising vaccines, and promising to, and basically paying these companies to start manufacturing the vaccines now, even before the clinical trial data comes out, such that if the vaccines prove to be ineffective or unsafe in the clinical trials, the government basically will have to pay for a bunch of vaccine stock that would have to be thrown away. But in the interest of speed and in knowing the production times it takes to get millions and millions and millions of doses of these vaccines out, the government chose to invest the money up front to get these things produced. And so that's basically where we stand today. The results that were published from which I have right here in front of me, I just got from a friend of mine at Moderna, that were published by the NIAID today, the Moderna vaccine are extremely effective. And I'll just give you a couple of these numbers right now. Looking at the first several thousand patients that were vaccinated, the primary endpoints of these particular studies was looking at patients at the number of actual SARS-CoV-2 infections, either through a clinical diagnosis, a CDC-defined diagnosis, or who are virus positive with symptoms. And also as a other co-primary endpoint, looking at number of cases of severe COVID. And there's certain definitions for what defines severe COVID from regular COVID. Just suffice it to say those definitions were done. And when they looked at the people in this first tranche of analyses, 90 patients in the placebo group got COVID. 11 cases in the vaccine group got COVID, 90 to 11. That's the 90% reduction they're talking about. I'm sorry, there were 90 cases in the placebo and 5 in the vaccinated group. Of severe COVID, there were 11 cases out of those 95 that I sort of mentioned. None were in the vaccine group. So 11 to 0. And so just to give you an idea of what's out there, again, only 5 of the 95 patients of the total people who got COVID were actually vaccinated, and none of the patients who got severe COVID from the Moderna, this is the Moderna vaccine we're talking about, got severe COVID. So very, very impressive numbers. And again, I think this not only, and there were no serious adverse events in this trial. So I think it basically shows that that these vaccines probably look like, I mean, these are still early data and they're not on millions of people, they're on a few thousand people, that there are no obvious glaring adverse events. And it certainly looks like there's a very robust immune response that's protective in patients who get the vaccine. So the data are shocking, I would say, just to say the least, and extremely promising. Just to round out, just for the last minute here before I turn to Jeremy, what other vaccines are in development? There's a vaccine from, there's a viral vaccine, activated vaccine that's the Oxford AstraZeneca vaccine we've all heard about. There's a vaccine that Janssen Pharmaceuticals is making, which is a piece of the virus inserted, piece of the coronavirus inserted into a non-pathogenic, what we call an adenovirus, that's then like the flu, some of the flu vaccines squirted into the nose, and then the adenovirus infects you, but you don't get sick. But the adenovirus now displays a coronavirus protein on it, so your body actually mounts an immune response to the coronavirus, even though you have a non-pathogenic adenovirus in your nose. There are a couple of DNA vaccines I talked about, which is from Inovio and Lineal RX. GlaxoSmithKline has another protein-based vaccine that they're testing right now, just the spike protein with an immune adjuvant, a thing that kicks up the immune system to that protein. That's a protein-based vaccine out there as well. There are 100 different candidates for vaccines, but the two we heard about this week that are the most easily to produce, most ability to produce the most rapidly in the highest doses are these RNA vaccines from Pfizer and Moderna, and those vaccines look very, very promising. And so with that, let me turn to Dr. Jeremy Rassen, not
that I can follow that.
epidemiologist extraordinaire. Well, Jeremy, you are really, I think, just to know, so Jeremy began his career, correct me if I'm wrong, Jeremy, because I know you both socially and a little bit professionally as well, having done some work with Aetion. But Jeremy was a Harvard-trained epidemiologist who did a lot of work sort of in medical epidemiology, basically looking at large swaths of data, clinical data, claims data, and stuff like that, and looking at trying to answer medical questions that way. A lot of times you can't do a randomized prospective clinical trial for everything out there, right? So if you want to ask yourself, is cough medicine associated with hip fracture, right? No one's ever done a clinical trial to show that my Robitussin causes a hip fracture or not, right? But you can go out there in the real world, pull billions of points of
record,
data.
that's hypothetical.
and you can answer that question, right? So talk a little bit about what you do, what Aetion does, and then we'll sort of put that in the context of the coronavirus.
So I trained in the field of epidemiology, in the subfield specifically of pharmacoepidemiology, which is looking at these kinds of data that you're talking about, John, to look at specifically safety and effectiveness of pharmaceutical medications. And the data that we use most often is what we call now real world data. We used to just call it observational data, and that was to distinguish it from, like you said, randomized data. So we work with non-randomized data, and you use these large non-randomized data sets, and you can observe in these data sets who takes which drug, who takes no drug, or who takes an alternative drug, and then what kind of outcomes do they experience. And then you can compare and contrast between the two. If you do that in a randomized trial, there's a process at the beginning, which is the randomization process, where kind of a virtual coin gets flipped, and you get drug A, you get drug B, you get vaccine, you get placebo. There's kind of a kind of random selection method of who gets what medication. In the real world, that doesn't happen at all that way. People get prescribed medications precisely because a clinician feels that they need them. So if you look at someone who's on a potent medication versus someone who's on a less potent medication, the person on the potent medication is probably going to have worse outcomes, even though that drug may actually work very, very, very well. And that's just because they're at higher risk of whatever this outcome is. So if you look at someone who's on a statin drug, they're going to have more heart attacks than someone who's not on a statin drug, just precisely because someone felt that a statin drug was appropriate. And so the work that we do uses these data, and the benefit of these data is that they're huge. Millions and millions and millions of patients recorded in these data. They're essentially real time, so you're not waiting for many years for a trial result to come in. And one of the benefits of the size of the data, aside from the sheer volume, is that you can look at a lot of different populations who couldn't possibly be addressed in a clinical trial, maybe even at all, or at least certainly in the numbers needed to really get a good understanding. So you think about older adults, you think about underrepresented populations, the numbers in a clinical trial likely won't really address all of those subpopulations in the way that you'd like them to. So these real world data can help you look both quickly, but also at populations that you could probably not even really observe in a clinical trial the way that you would want to. You can also answer questions that you couldn't design a trial to ask, because the trial, if you could run it, would be unethical to run. Maybe you're looking at drugs where you couldn't actually randomize someone from one drug to another because there wasn't what's called equipoise, where there's kind of scientific what's the right word? Sorry, it's 8:30 here.
We have that in rheumatology now. We've come up with such good therapies that we no longer can study new therapies against just a placebo, right? And so it's very different. So you have to either add it on to existing therapy. It makes the clinical trial much harder to do that.
And then when you're thinking about safety, these trials that we're talking about for vaccines have tens of thousands of patients in them, which is huge. I mean, these are huge trials, and the Pfizer trial, I think, has about 45,000 patients in it, going down to age 12. But we're talking about safety events that could be 1 in 100,000 or 1 in a million or 1 in 10 million. And the statistical likelihood of seeing one of those events in a trial that's even as large as, call it 50,000 people is actually relatively low. So these large data sets can really help us understand safety on a much bigger scale, and extend that understanding of safety to rarer outcomes, which probably won't be observed in a trial. So we do this every day. We do this for a lot of different medications. And as John said, we work with manufacturers of these medications. We work with payers who are looking to understand the effectiveness of the medications, maybe not in a general population, but in the population that they serve specifically. And then we have, as John mentioned, at our company, a research collaborative agreement with FDA specifically around COVID-19, understanding the disease, understanding the treatments, and understanding diagnostics, and hopefully we'll have some data soon, very soon, to start to be able to understand vaccines as well.
So that's a great overview. I mean, I think that the thing that I think people should really remember is you're exactly right. There's no way to do a clinical trial in millions of people. The costs are prohibitive. And also, I mean, as someone who does clinical trials for a living myself, I'm running 3 clinical trials right now. I can tell you that clinical trials are not the real world. Patients are not selected at random. You're actually right. You're absolutely right there. They have to meet very, very specific eligibility and inclusion and exclusion criteria. And patients are followed in a way that the real world doesn't really match very well. For example, I had a little procedure on my neck today. I'm supposed to take an antibiotic 4 times a day now for the next week, right? While I do this, there's no way I'm gonna succeed, I think, in taking every dose when I'm supposed to take it, right? But in a clinical trial, for example, you show up, you have to bring your pills with you, and the study personnel count the pills and we'll give you phone call reminders and follow-up reminders, and it doesn't mimic exactly what happens in the real world. So in order to get real world numbers about what's going on out there, you're right, you can't look at clinical trials. You have to come up with more innovative ways to interpret the data that is out there, whether it's through medical claims that people make, hospital records, insurance company records, pharmaceutical company records, clinical trial records, all together. And I have to say, it seems so, easy on the top to actually to sort of talk about that. But how in the world do you develop a computer algorithm, Jeremy, that could actually do that and adapt in such a way that you can answer a question, for example, relating to COVID? And we'll get into COVID here in a second.
I'll say transparently. And the trick here is to not have any tricks in the algorithms. We want to be able to do these analyses in a way that anybody who's looking at it can understand what we did, why we did it, and how we did it. And that's because these analyses have kind of so much real world meaning and real world impact for patient health, that doing it in a way that anybody who's looking at it can understand what was done and why it was done is a really important part of the work that we do. So, you know, we have in our product technology for doing it quickly, for doing it replicably, for doing it in a way that kind of meets kind of standards around how it should be done, but the actual kind of mechanics of the analysis itself, we do completely transparently. And that's really important, especially as we're talking about vaccines and other products that will be used, you know, by millions and millions of Americans in all likelihood, right? And we have to be thinking about transparency and interpretability of those results.
So let's turn our attention to the COVID vaccine right now. So so just to COVID in general. So, you know, there's been so many questions about COVID. Like, you know, is it real? How is it being spread around the country? Are there super spreaders? Is one person responsible for 20 infections in an area, or 20 people responsible for 20 infections? And 19 people who have the infection don't spread it to anyone else. Is it more severe now than it was? Is it less severe? Is it more you know, communicable now than it was previously? How do we answer these questions, Jeremy? And have you been, has Aetion been used in such a way by various government agencies and other organizations to try to answer some of these questions? And if so, how have we tried to answer them?
Well, I mean, I think understanding a lot of those things you mentioned, you know, we put that under the heading of natural history of COVID, and how many patients are being diagnosed? How are they being diagnosed? Is it through lab tests? Is it through you know, clinical observation? What is the trajectory of patients' health once they're diagnosed? How many patients go into the hospital? Once they're in the hospital, what medications are being used? How well do those medications work? All of these are actually, the last part is not natural history. That's a comparative question, but all of these things are things that we understand, and we can do this now looking retrospectively, you know, 6 or 8 months in the data. And one of the things that we've worked very hard on in this era is getting data that's no more than kind of a couple weeks old, because this is moving very, very quickly. The treatment patterns are changing very quickly. New drugs are arriving, new kind of methods of treating patients within hospitals are arriving, and being able to understand not just what's happening kind of on average over 6 months, but month by month, or even kind of more quickly than that, has been a really important part of the work that we're doing. We have a graph that we publish on our website of medication use, and maybe I can just pull this up here and show it even.
I may have
it's
fascinating.
Yeah,
so
it's
medication use over time, and you can see on this graph, it's amazing. You can see the rise of the use of steroids, for example, and a drop in the use of certain other therapies that were sexier. Hydroxychloroquine, there you go. Oh,
yeah, and so you can see this week by week. So hydroxychloroquine, I think, is the red line, yeah, and you can see that this goes down over time, pretty precipitously. You know, March into April, and then the blue line is dexamethasone. And if you remember, over the summer, early summer, there was a big trial result out of England talking about the effectiveness of dexamethasone, and you can see how, like, almost immediately that gets picked up by hospitals, by doctors here in the U.S., and that gets used on patients, and that number has, you know, stayed pretty high. This is 50% of hospitalizations at this point include some use of dexamethasone, and then the purple line is other steroids. So you can add those together even.
These data are amazing, Jeremy. So just to highlight this out there, and you answer this question: how do you get these data? There's no one central database that reports medication use amongst the 50 billion hospitals in the U.S. As a matter of fact, the government has actually obfuscated some of these data by taking some of the data away from the CDC and firewalling it within Health and Human Services. So tell us how are you able to aggregate all this data from various big hospitals, small hospitals, public hospitals, pharmacies, elsewhere out there? Is it pharmacy data? Is it hospital data? Is it medical records? And how are you able to get it so fast in such large numbers?
So it's all of the above. And here, just before I close this, this is remdesivir. You can see the rise of remdesivir over the last 4 or 5 months or so. So it's all of the above. And I should start by saying that all the data we work with is fully de-identified. And I know that that's that's a bit of a buzzword in some ways, that of course the data is anonymized, we don't know anybody. Here it's de-identified, and there's actually a statistical certification process where somebody looks at it and says, not only is it de-identified, meaning it doesn't have the identifiers that you use to identify someone personally, but it's also kind of broad and sort of fuzzy enough in certain ways that you couldn't implicitly identify someone if you tried to do it. And that goes through a statistical certification process. But to get all these data together, we work with all of the above. So insurance claims are a major source of data. So when you see a doctor or go to a hospital, a claim gets submitted to the insurance company. That claim has the procedures that were performed, the diagnoses that were diagnosed, and you can get some information, not super detailed clinical information, but some information from those insurance claims. You can add to that hospital billing information. So when we think about which medications are being used in a hospital billing systems can provide that, because every medication that gets used gets billed. And we can look at that information. We can look at medical records that have been as I said, de-identified and certain fields pulled out of there for clinical context. And then pharmacy claims as well. So you put it all together, you get a picture of outpatient medication use, inpatient medication use, outpatient procedures, inpatient procedures, and you can start to see some of those questions you were asking before about natural history. Well, how does a patient who's diagnosed in an outpatient setting, who gets admitted, what happens in the hospital, what happens once they're discharged, and really kind of understand that full picture over time. It's pretty interesting stuff. As you can imagine, there's a lot of detail to it. Like, one of the things that we think a lot about is, what are we seeing in the data, which is obvious in some ways, but much more subtly, what are we not seeing in the data? What is not being recorded? What is not being included in these data sets? And if you try to make kind of a conclusion based on what you think you see, but you're not really sure, it's important to understand kind of the denominator, if you will, of what's not being included in these data sets. So
that's a great explanation. I think, have you done any forward modeling based on the data you have, the trends that you see now, to sort of look at what future medication use is going to be? And have you modeled it to see what the effects of certain medications have been on clinical course, like mortality, severity of disease, things like that? And is part of the reason why we're seeing fewer deaths now related to the change in medication use? Did you see some of that in the data?
So there were a couple questions in there. And so in terms of forward modeling and making predictions, we don't do a lot of predicting. Or I personally don't do a lot of predicting. I mean, you saw those plots, and you saw, one news story can change, a drug from being, down here on the plot to being all the way up here. And so, predictions are a little bit, let's say, challenging. But, we can use the data to understand which treatments are more effective, but also which combinations of treatments, or which treatment given in a specific setting is more effective. And so you can really tease out a lot of information and use that inference to help clinicians who are making decisions kind of minute by minute to make the decisions that will likely have the most benefit for their patients.
Have you looked at timing of medication use as well? So, for example, like in the remdesivir story, that if you wait too long, right, it probably doesn't work very well. There's a certain time when you want to use it, and it's maddening because it's a very expensive drug. You have to be admitted and get it intravenously, but the time to give it is when people really aren't that necessarily as sick as they may or may not get into the future. So, have you looked at that modeling in terms of the timing when it was used previously and how it's being used now?
Yeah, I mean, timing is something that we've looked at, maybe not specifically in that question, but understanding when in the course of a hospitalization a drug gets used. You're the expert here, but on the flip side of that, if you use dexamethasone too early, you can be creating trouble that you didn't have to start with, right? And so understanding timing in general, timing relative to kind of a sentinel event like a positive test or a hospital admission, or something where you can sort of have a reference point in time to compare against, something that we do a lot of.
Can we go back to that graph for a second? I want to point out a couple other interesting things. I remember looking at this graph earlier.
All right, I got to get the graph. I got to get the graph.
I always followed what Aetion does because I think the data you guys get, again, the rapidity that you get for this, how quickly you can produce these data are really.
You can see here that our partner here in data is a company called Health Clarity. They're based in Philadelphia.
These are amazing. So in addition to seeing specific viral treatments like remdesivir that you can sort of see in here and hydroxychloroquine, what's interesting is I noticed that you have anticoagulation in here as well. So you've got both the use of heparin, which is an intravenous thing, and given intravenously, and low molecular weight heparin, which is something we inject. And it's been learned more and more as time goes on that part of the severe complications related to COVID are the fact that people get clots in their body, in their veins, in their arteries. They get strokes, they get heart attacks, they get pulmonary emboli, things like that. And it's interesting, if you look at your use of low molecular weight heparin, it clearly goes up right around sort of in the middle, like in June or so. And it's generally held steady, as has the use of unfractionated heparin. I think so. This gets a little bit technical. I work in a hospital, so I can read into these data a little bit. We always use prophylactic doses of these drugs, right? In people who are hospitalized and immobilized. That's been standard of care now for a long period of time. Can you tell the difference between prophylactic dosing of these drugs and pharmacologic dosing to actually really do, either treat or what the doses we would use, for example, in COVID, for example? Can you tease that out?
We can. In this case, we didn't distinguish between lower doses and higher doses, but we can see the difference between the two. I'll just point out also the black line at the top here, this is Tylenol. And so we included this because we wanted to, that's kind of our reference. And we expect that Tylenol over time should be pretty much flat, and that's what we're seeing. And so that's our reference drug in here.
Well, it's beautiful. The data are absolutely amazing and beautiful, Jeremy. Let's talk vaccine for a second. Have you guys been recruited in sort of so we got a lot of questions about how a vaccine would work, how it'd be distributed. We all have heard, for example, number one, that the problem with one of the shortfalls of RNA-based vaccines, unlike protein vaccines or virus vaccines or whatever, is that you have to really refrigerate them. They have to be refrigerated at minus 80 degrees. And I have a minus 80 freezer in my lab, but a lot of people don't have minus 80 freezers lying around.
I thought you were saying your basement.
No,
with a porch or something. I don't know what you put in that minus 80 degrees.
My mother-in-law, maybe. Just kidding, Mom. We actually have, nobody has, minus 80 vaccines, minus 80 freezers lying around. It's very, very difficult to procure. They're tens of thousands of dollars. Have you bothered to look at the distribution networks in the United States? How is this vaccine going to be distributed? What's the most equitable way to get it out there and sort of modeling, but actually looking at viral transmission to see what are the most effective ways to sort of get this vaccine out there and reduce the viral transmission?
I'll be honest, John, that's not something we've really looked at. What we're thinking about is, given the distribution plan and the CDC on their website, they have a distribution plan. It's phase 1A, phase 1B, phase 2, phase 3, and it's kind of everything you've read about, priority populations, healthcare workers, older adults in nursing homes, those at substantial risk in phases 1A, 1B, and then, essentially everybody else in phase 2 and phase 3, with people who want it earlier in phase 2 and people want it later in phase 3. And so what we've been thinking a lot about is where will the data come from that help us understand safety across all of these phases, and given the distribution strategy, how that will all come together. And so that's where we've really been thinking about distribution, really in the context of how we can observe safety given the plan of the rollout. And if the rollout plan had been different, and I'm not talking policy here, I'm just talking about being responsive to what the plan is, right? If the rollout plan had been, we're going to equally distribute the available doses randomly across the country, that implies one strategy for understanding safety, but rolling out to specific target populations, that implies another strategy for understanding safety. Not so much the statistics or the mechanics of the statistics, but where are the data going to come from that are going to reflect those populations, so that we can really understand that in essentially near real time.
So let me rephrase what you just said for everybody out there, because I think this is really, really important. And this is where I think, Jeremy, you're going to play a huge central role in this. Everybody's afraid for the safety of the vaccine, right? Are these vaccines safe? You know, we've never used a vaccine like this before. I don't know, blah, 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 whatever. These trials only have a few thousand, you know, even 10, 20, 30,000 patients in them. If, you know, you have a 1 in 100,000 patients becomes paralyzed from myelitis of the spine, for example, you won't see it on the in a clinical trial on the law of averages, right? So how would we ever know? We're going to rely on you, right, and Aetion to actually, when this vaccine starts getting out into the public and you start getting hundreds of thousands and millions of doses of it administered, you're going to be getting the real-time or near real-time data out there. Your sensors and your computer algorithms will be out there gathering all this data from around the United States and looking to see if there really is a post-marketing signal out there. So we will know relatively, we will have the mechanisms in place to know quickly, even without a clinical trial, if there is a safety concern, and we can raise it before more people would get harmed, right?
And just to be clear, FDA will be doing the same, others will be doing the same. This is something that, you know, everybody in the field I've talked to is taking incredibly seriously. And I think it's you know, it's a pretty much all hands on deck situation for making sure that any vaccine that comes out under an emergency use authorization, which is the first step, or a full license, which is the second step, will have you know, a total understanding of safety, both from the clinical trials, but then also as it's used in real world populations. And there was a really interesting meeting that happened, oh gosh, maybe 3 weeks ago, 4 weeks ago, which was it's called the VRBPAC meeting, the Vaccines and Related Biological Products Advisory Committee meeting, which was a 8 or 9 hour meeting that happened virtually. It normally would have happened at FDA, but it involved FDA staffers, representatives from some of the industry players like the vaccine manufacturers, and then members of the public, and then members of an advisory committee that the FDA has, a standing advisory committee to think about vaccine safety. And a lot of really interesting questions came out of this. This was before the Pfizer numbers, before the Moderna numbers. And what the committee, you know, what the FDA was saying to the committee, and I think you've seen this in the news, this was a fully public meeting, was that they would think about an emergency use authorization. So that's step one. So to get a full licensure, there's an application that's, you know, I don't know how long, thousands of pages long, that includes all kinds of technical detail in it. And it's really important technical detail, but it's technical detail that, you know, may take longer than we want to wait as a society for a vaccine to come together. So they're looking at an emergency use authorization to come prior to that, where they said they would put a threshold of 50% effectiveness, with a lower bound on the confidence limit. So, you know, when you're talking stats, you never know precisely what you're talking about. You're talking about a number which has some uncertainty to it. It's like the margin of error in a poll, where they said at 50% effectiveness with a lower bound of the confidence limit at 30%, and thus, you know, implying an upper bound probably somewhere around 70%, and then also requiring a certain number of cases to have occurred. And they require the certain number of cases to have occurred because they don't want the vaccine to look effective just because there was no virus in the trial population. So they put that criterion in there as well. And a lot of discussion about that. But I think, you know, the 50% number, I think people were, you know, I think we'd heard that 50% number for a while. The other piece that was in there, though, was how long will they have required that the patients have been in the trial have been followed for potential safety signals at the time of the emergency use authorization. So the number that was there was 2 months of median follow-up after the second dose in the trial, which means that, you know, half the patients had to have been followed for at least 2 months after the second dose for the EUA to be considered. And so, you know, that's a substantial amount of time. And John, again, like, jump in here because I don't know the precise details of this, but a lot of the adverse events that you would expect to see as the result of a vaccine happen fairly quickly, certainly within 2 months. You know, I think 4 to 6 weeks is, you know, roughly the amount of time where the majority of adverse events would happen. So 2 months should be enough, but a lot of people are saying it's something, but it's not as long as we want to watch for. And so certainly the trials will continue, but let's come back to that because that's a really interesting point as well. The trials will continue after the emergency use authorization. The patients will continue to be followed for potential safety problems, and there's be plenty of time, years of follow-up to be included. But we can, as the vaccines are used across the country, we can use the kind of data that my company works with, real world data, to understand safety in larger numbers, and then as time goes on, over longer time periods as well.
You're exactly right.
As I said, the trials don't stop at the time of the EUA; they keep going.
That's exactly right. You're exactly right. Most of the side effects would be within the first few weeks of getting the dose. It's less of a concern with the RNA vaccines theoretically, although this is a lot of this is the hype behind the technology, but the theoretical risks of the RNA vaccine, or the RNA makes its transcript, and your cells express it, and then it's gone. It degrades and it's gone. A lot of times when you give either a live or killed virus to someone as part of a vaccine, or you get infected with a virus, you sort of get post-viral immune reactions. Guillain-Barré syndrome and things like that can occur, in the weeks or so after a recent, diarrheal illness or viral illness or whatever. So less of an issue with the mRNA vaccines, and theoretically an issue that you would see closer into administering the vaccine. So you're exactly right. But oh, look at this! This is great. So explain this to the audience out there, Jeremy.
So I'm sorry, I haven't been watching the chat all that carefully, but I saw a question here about essentially the phases of rollout. This is from the CDC website. This is the COVID-19 vaccine program interim playbook, and you can see those phases that are talking about phase 1, phase 2, phase 3, with phase 1 divided into phase 1A and phase 1B. And David, I see your question as well, and I have a fun answer for that. So you can see where different groups would fall in these phases. I'll just leave it up here for a sec. There was an interesting issue that came up at this meeting that sort of caught my attention. It's almost Talmudic, if you will, Jessica, in that I'm going to put this down so I can see everybody, which is that there's this, we talked a little bit about equipoise, and equipoise is something that's an important part of clinical trials, where it says it's only ethical to start a trial if you're in this state of equipoise. You truly don't know whether the drug you're testing is better than the alternative that you're randomizing, call it half the patients to, right? And so, at the beginning of a vaccine trial, you're in equipoise. You don't know whether the vaccine is going to be better than placebo. That's kind of the state you're in. But the ethics say, if you believe the agent that you're studying to be highly effective, then you need to stop and say, we're no longer in equipoise, right? And and it can work the other way as well if you think it's harmful, and say, we need to make another choice here. And so, what came up at this meeting was that if you think about what does getting an EUA mean, an emergency use authorization, it means that the vaccine has been tested enough such that it is believed that it is at least 50% effective with all those conditions that I mentioned. And at that point, are you still in equipoise? Are you still in a place where you think that placebo is probably, or is as likely to be as good as vaccine? And the answer is probably no, right? Because you've made the statement that you're getting the EUA because the vaccine appears to be working. And so what some of the manufacturers were saying, or at least there was a couple of the manufacturers at this meeting, and what they were saying was that they think that at the time of the EUA, they have to unblind the trial, because trials are blinded. So you don't know whether you got a vaccine or a placebo. And you do that so that people who, for example, know they have a vaccine, want them to take part of riskier activities, going out and hanging out with a bunch of friends with no mask, because they think that they're, vaccinated in some way. That can kind of throw off the results of the trial. So patients are blinded. You don't know if you get vaccine or placebo. But they said at the time of the EUA, they would have to unblind the trial and offer patients who had gotten placebo the opportunity to get the vaccine. And so at that point, you have what's called a huge crossover in your trial, where patients cross over from the placebo group to the vaccine group. And at that point, your randomization ends, because the patients have made the choice to move from one group
Exactly.
to another. And you have to think about how you continue to understand safety long-term in the trial once there's been a large crossover from one group to the other, and that crossover was likely non-random in some way. So, from the public health point of view,
well, we've seen that. We've seen that with remdesivir, right? Once remdesivir got its EUA and the early studies came out suggesting it had some efficacy, right? How could you randomize somebody who's sick to placebo versus remdesivir, right? Everybody started crossing over into the active group. So we need you to go look at the real-time data as it's coming in, as it's being used in the real world, to answer those questions about safety versus a theoretical placebo, right?
So the public health point of view is that, continuing to participate in the trial in a blinded way improves everybody's health, or kind of the general health, in such a way, because we learn from the continued experience of the patients who are enrolled in the trial. The medical point of view says, no, it's better for the patient probably to cross over from one group to the other. And that's sort of the fundamental tension between medicine and public health. And, John, you and I can duke it out later. I just know you'll throw me in the -80 freezer, and that'll be the end of it.
I'm a purist like you. No, I like good, pure clinical trial data. It's science to me, exactly.
But that's the Talmudic part of it, if you will. Like, kind of what's the right answer there? I don't know. I'm no expert at this, but it feels like there isn't a single right answer to be had.
So that's beautifully described, Jeremy. It's the best description I've heard yet of really why we need to have ongoing surveillance, even while the clinical trials are ongoing, or even after they're over, to try to answer some of the questions that can't be answered in the clinical trial, both because we can't ethically randomize people evenly to placebo or drug, because we now have a bias versus that the vaccine, for example, works, and also because the numbers just won't be there, at least early on in the pandemic.
Can we answer this question that David put in the chat?
Yeah.
Can we better prepare for the inevitability? Whoops, it just scrolled. Better prepare for the inevitability of new viruses. So I was involved in a small part of a very large effort that was led out of UCSF to look very specifically at this question, and it was a really fascinating research endeavor. It was published in Science Online recently, and I think will be in the printed magazine at some point soon. And the question was, what can we learn? And by we, I mean the collective researchers, not including myself, because the lab part of it, not my area. But what can we learn from SARS-CoV, or original SARS-CoV, SARS-CoV-2 and MERS at a molecular level? To understand, what are the shared properties of these three viruses? What kind of drugs would we need to have in order to block the activity of these viruses? And do we have any such drugs? And the paper looked at the molecular structure and understood kind of all the mechanics of it, and then postulated that there are a couple groups of drugs that might actually be quite effective, and we were able, and this is the part where my colleagues and I came in to test those hypotheses using the kind of data that we have. So we looked at a drug called indomethacin, which again, John, a rheumatologist friend, can tell us more about, and looked at typical antipsychotic drugs as potential.
Olanzapine. Was this the Pasteur study with the Pasteur Institute and UCSF?
Pasteur, yeah.
You were part of that study? I didn't know that. That was a huge study that got published in Science a couple months ago. Wow.
And so, coming back around to the question, here was, Nevin Krogan, who was the PI of that study, when he was talking about it, he said, yes, this is about understanding how we treat SARS-CoV-2, but because we're building our understanding on SARS-CoV-1 and MERS and now SARS-CoV-2, it'll help us predict how we're going to be able to treat SARS-CoV-3 when that, perhaps inevitably comes along at some point. So that was a long way of answering a short question, but hopefully
basically
informative in some way.
So this was basically repurposing drugs in existence and chemical compounds known to exist in the vaults of pharmaceutical companies that may not even be clinically used yet. We're able to show that many of them had both theoretical activity in vitro, in the laboratory activity, and then with your data, real world potential activity as well. And that can be applied, using this as a framework, can be applied to future infections as well. It's really,
and I think a lot of it was about like, what are the structures of a drug that would be needed? And we have some drugs today that have those structures, but they're also for other things, like typical antipsychotics weren't created to fight any coronaviruses, certainly. But we can use the understanding of whether that property of typical antipsychotics is effective in reducing the severity of SARS-CoV-2 to design a drug that has this anti-SARS-CoV-2 property, but not the antipsychotic property, which would not be wanted certainly in the vast majority of patients. So it's sort of an interesting way of using what we know today about existing drugs to think about what new drugs would need to look like to fight both this pandemic and future pandemics as well.
And I'll wrap up just by also saying, to answer David's question a little bit, these mRNA, part of what makes these mRNA vaccines so promising, David, is that it's plug and play. It's the ability, if you get another emerging infection causing a pandemic, in the future, it's the ability theoretically to take genetic code from that virus that codes for a region of some novel virus that's mounts an immune response, to be able to take that, plug and play it into the Moderna cassette or into the BioNTech Pfizer cassette or whatever.
Showing your age, John.
Generate a brand new vaccine. I mean, that's the ability to do those things for future pandemics.
Into the Moderna phonograph, at least.
That's right. Well, Jeremy, thank you so much. This has been just an incredible discussion. It's probably, I've loved all the ones I've done so far, but this one ranks up there as one of the best. It's just been a pleasure to see you again and to engage in this very, very wonderful discussion and share all this information with our wonderful congregants out there. And I'll turn this over to Jessica now.
I just, I'm turning the computer here because we're showing one computer tonight. I want to say that I feel, what Jerry Flanzer just said in the chat, I feel really, really hopeful. And the idea behind this from the front lines conversation was to take this incredibly complicated and confusing time in our lives as we navigate this pandemic and really come back to our closest friends and allies, and especially people who are really part of the Share with Israel community, to try and get the kind of information and from the front lines to understand really what's happening out there from our friends. So to Jonathan Graf and Jeremy Rassen, two very good friends, thank you so much for being with us and for being back at Share with Israel. And Jeremy, I hope that you will come back again to talk with us about sort of the way this vaccine situation unfolds and what we're seeing as you continue to research all that's going on out there and changing quickly.
And I think it's one of the standard things we say these times, which is that, I wish this could be in person. I certainly wish this could be in person, but also, I wish this could be at Share with Israel, a place where I spent a very substantial part of my formative years and have a really close connection to. And next year together and at Congregation Share with Israel. So thank you, Jessica, Rabbi Graf, for the opportunity. I really, really appreciate being able to be with everyone tonight.
Well, we're so happy to have you and so happy to have you doing this work out there on behalf of the entire world and especially this place where you grew up. Shout out to your parents who I guess you said are on here. I can't see everybody who's out there, but to Amy and Josh Rassen, thanks for having us.
And to yours as well. I saw your dad too.
So thanks everybody for joining us.
And to my mother-in-law as well. Just a joke.
Yes, next year in person, soon in person, and see you all Friday night at 6 o'clock. Jeremy, you should know that Laura Manus is on from our Sunday school class as well.
I saw that, and Rebecca too was on.
Is Rebecca Handler on? I didn't.
She was.
Congratulations to Rebecca Handler, who just published a book this week, and we'll look forward to having her to talk to the.
Has everyone pre-ordered Rebecca's book?
Thank you all for joining us. You should, you must. So there's Laura. Many more cool things happening from our class out there to be continued. Thank you. Good night.
Bye, everyone.